Hello, and welcome back to Honors Biology. So today we're gonna talk about mutations and then kind of towards the end talk a little bit about gene regulation and how often different genes are expressed, just briefly at the end. So let's get into mutations. So a lot of you have been kind of asking about mutations. Well, what happens if this process doesn't work right? What if this changes? What if that changes? All of those questions are mutations, and I've been putting off answering them because this should answer most of your questions. So what mutations are is they're changes in the genetic material. So they're changes in either the DNA or the RNA for gene mutations. Usually they're going to occur during DNA replication or transcription. So either one of those things can happen. So like example, this frog here is having some problems with his legs because his cells in his legs have his cells have a mutation that causes that kind of protein to develop legs to grow where they shouldn't grow. So there's two types of mutations. There's chromosomal mutations, which involve the number or structure of chromosomes. So if you look at that picture, most chromosomes, all your chromosomes come in pairs. But in Down syndrome, we call it trisomy 21 because for the 21st chromosome, people who have Down syndrome have 3 copies of the 21st chromosome. We'll talk more about that when we get to the cell cycle and karyotyping because there's a whole, set of mutations and kind of disorders that go along with that. The other type that we're going to focus on right now is gene mutations. Gene mutations are mutations that produce change in a single gene. So gene for eye color, gene for skin color, whatever. Here, this turtle's having some problems with its shell. It grew 2 shells because a change in the DNA at that gene produced a protein that caused an extra shell to grow. So let's talk about gene mutations. Gene mutations fall into 2 categories. There's either point mutations, or we call them substitution mutations, or frame shift mutations. So we're first going to talk about point mutations or substitution mutations. Substitution mutations are just when one nucleotide gets replaced with another. It's one nucleotide gets substituted for another. So example here in sickle cell anemia, this is the normal DNA sequence, CTT, but due to a point mutation, this T gets replaced with an A, which causes then the mRNA to change as well when it gets transcribed. And then when the mRNA gets translated, it produces a different amino acid here, which changes the hemoglobin. So hemoglobin is the protein that binds blood or binds oxygen in your blood. So because of this one change, we get a different amino acid, which completely changes the structure of the protein. Remember, structure determines function here. So we change the structure, we're going to change the function of it. For point mutations, we have 3 different types of point mutations. The first is called a silent mutation. So a silent mutation is when the base is changed, but the new codon that appears codes for the same amino acid. So there's really no effect on the protein that gets made. Usually going to occur in that third letter. That third letter of the codon is kind of what determines what amino acid gets made. So, for example, here, this is our original RNA strand with the sequence. This is the changed one. So here, there's a U here instead of a C. So there's a U instead of a C here. But we're still gonna produce glycine. And that's because there's more than one way to make glycine out of a codon. Some of you were asking, right, the codon chart, the DNA code is redundant. There's a lot of different ways to get a lot of different amino acids, right? There's 6 ways to produce leucine. Most of them have at least 2 to 4 different ways to produce amino acid. Like proline, as long as you get the first 2 letters right, it doesn't matter what the third letter is, you're going to get proline. It's redundant to help fight mutations like this. So that way, if a substitution mutation does occur, it's not going to affect anything because you're still going to get the same amino acid. So you're just kind of increasing the odds that you're going to get a silent mutation. So the second type of point mutation we can get is called a nonsense mutation. A nonsense mutation, it's when if there's a change in the DNA that causes the protein to stop earlier than expected. Let's call it a stop mutation. What that looks like is here's the normal sequence of mRNA to proteins. Here's the nonsense mutation. So this A got replaced with the U. Well, now instead of AAG, which codes for lysine, we have UAG, which is a stop codon. So instead of all these amino acids getting made, this protein gets made, we make the first amino acid, methionine, and then it stops. So then the protein doesn't get made, or it gets cut off, so it's shorter than expected, which then it doesn't work right, is the function isn't there. So that's a nonsense mutation. The third type of point mutation we can get is called a missense mutation. So a missense mutation is just when the nucleotide changes, it gets subbed out, swapped out, and then the code just changes one amino acid. So what that means is this is the normal type here, this is the mutant type or mutation. So this T got substituted or changed to an A. So instead of cytosine, we get serine. The rest of the sequence is still the same, right? All the other three amino acids still get made, and they're still the same amino acids. But because we changed that one, we then run the possibility, we have a high risk then of the protein's function changing because the structure is going to change. So changing one amino acid can change the structure of the protein, which is then going to impact how it functions. So those are your three types of point mutations. You have missense, nonsense, and silent ones. So those are your three point mutations. Your next group of mutations are called frame shift mutations, and there's two of them, deletion and insertion. How frame shift mutations work is when we set up the codon, I told you we have what's called a reading frame. We have to identify the reading frame. Well, the reading frame can get changed. It can shift depending on if we delete or insert a base. So if we delete a base, one of our A's, C, G's, or U's, we then change the whole reading frame. So example, the classic bio example is the fat cat ate the rat. Groups of three, wonderful. But if we delete this H low, we then change the whole reading frame because we have to have these groups of three. We have to. So then it becomes this TIF act H tit her, which is completely unreadable. It makes no sense. So in a deletion, you can have, so all of these are going to produce different proteins or different amino acids than what was originally made. So you may get an entirely different protein to occur, or the protein just may not occur. It depends on where the deletion occurred. And then the other frame shift mutation we can get is just an insertion. So a base gets inserted in between two other ones. So which causes another shift. So for example, and here's our original strand here, and then here's our mutated one. We inserted one. The insertion occurred in between the A and the C here. Well, then that shift everything over because we needed to have groups of three here. So instead of this being our wonderful amino acid sequence, we then get this, which in all reality probably wouldn't even get made now because we don't have methionine, we don't have that start codon. So frame shift mutations still result in proteins, amino acids being made, but we change the reading frame. We change the sequence of groups of three that gets made. So there's your difference between point mutations and frame shift mutations. Point mutations just affect one nucleotide. We just swap out one nucleotide base, and that may mess up the protein. Frame shift mutations, we actually change the reading frame, how the amino acids get read is changed, how we group them and the codons get changed. So there's your difference between point and frame shift. So make sure you keep those two straight. Now, not all mutations are bad. And the reality is most mutations don't have any effect on gene expression or how proteins are made. Because I said before, most of your DNA, 90% of it, doesn't code for anything. So that means probably right now you have mutations in your DNA, but it's in a non-coding section of the DNA. So it doesn't matter because it's not affecting anything. But if they occur in a actual coding part though, then we get some problems. We can get different genetic disorders to occur. We can get cystic fibrosis, Huntington's disease, hemophilia, Duchenne muscular dystrophy can also occur. Most times, if you have one of these, and they're super rare, if you have these rare genetic disorders like cystic fibrosis or Huntington's, they most times are fatal. Most people don't survive them because your body actually isn't functioning the way it should function. Cancer can also arise as the cause of through mutations, and we'll talk more about cancer when we get to the cell cycle because it's part really cancer is unregulated cell growth due to mutations. But mutations can also be good. When we get to natural selection and we talk about differences in species, genetic variability in species is really beneficial to species survival, right? Skin color is just different amounts of a protein called melanin that gets produced. So if you have a darker skin color, you have higher amounts of melanin in your skin. If you have lighter skin color, you have lower amounts of melanin gets produced. Higher amounts of melanin is super critical if you live close to the equator where there's a lot of sun because that protects your skin then from the sun's rays. If you live where there's not a lot of sun, it wasn't important. So those evolved to help animals of different species survive. So not all mutations are always bad. Sometimes it's really good. And the different eye colors, blue eyes, green eyes, brown eyes, those are all mutations that arise, right? So some of them just change the appearance. Eye color, they're all different mutations and just there now. Um, Briefly, just real quick on gene regulation. So briefly talked about this, but not all genes are actively expressed all the time. It's not beneficial to the cell. Really, it's just the proteins that need to get made are the ones that are being made. So, and it's just really a time thing because you're wasting energy if you make all of them in raw materials. And also, it's really bad for your body if certain proteins get made kind of throughout when they're not supposed to be made. That's what Huntington's and cystic fibrosis is these proteins get made constantly that don't need to get made. And then we can have some really bad consequences from that. So there's some genes that get switched on and off, and it just depends on the situation. Some genes we get rarely expressed, like adrenaline. Adrenaline is a protein that causes dilated pupils. Your heart starts beating faster. It's kind of that flight or fight response kicks in. That's adrenaline. That's rarely expressed gene. Only in certain situations does your body produce adrenaline. Right? You don't have adrenaline coursing through your body right now. It's not useful. Other ones like hair growth or blood pressure, those get constantly expressed. You need them every point of throughout your whole life. So those are always being expressed. And then some genes, they're expressed for a time, but then they get turned off. So the genes for going through puberty, estrogen and testosterone, those are only expressed when you're going through puberty. And then when you reach maturity, when you're done going through puberty, they get turned off. There's other genes that get expressed just different parts of the day. There's a gene called melatonin, which is the drowsy. So if you get real drowsy at nighttime, your melatonin kicks up. But throughout the day, you have low amounts of melatonin, and at night it increases. So it just depends on what time of day that different kind of genes get expressed. And then thirst genes can also be influenced by environmental factors. So temperature can cause genes to be turned on and off, or mutations to occur. Light, too much sunlight can lead to skin cancer, which is the sunlight, the radiation from the sun causes a mutation in the skin cells to become cancerous. It's a big concern when women are pregnant to make sure they don't smoke or drink or do drugs or anything like that, because that can negatively impact the child, because that can harm the DNA and the development of the child by inducing mutations. So have to be careful. So that's all I got for today. If you have questions, let me know in the video. Other than that, I will see you in class.